to another episode of BioVlog. This video will focus on the discussion of cell cycle and cell division, including the processes and the stages of cell cycle and the types of cell division. Without further ado, let's get started! Cell cycle and cell division. Cell cycle is the ordered sequence of events that occur in a cell in preparation for cell division. This process takes place in a cell as it grows and divides. The cell cycle referred to as the life of a cell. The cell cycle has different stages called G1, S phase, G2, and the M phase. It has 4 stages process in which the cell increases in size. The stages G1, S phase, and G2 make up interphase, which accounts for the span between cell division. The 4 stages in cell cycle. First gap. It is the first stage in the cell cycle. It is the stage where the cell is preparing to divide. During this phase, the cell grows in preparation for DNA replication and certain intracellular components such as the centrosomes undergo replications. The S phase of a cell cycle refers to DNA synthesis. This stage is responsible for replication of DNA. In this way, the genetic material of a cell is doubled before it enters mitosis or meiosis, allowing there to be enough DNA to be split into daughter cells. The S phase only begins when the cell has passed the G1 checkpoint and grown enough to contain double the DNA. The second gap, or namely the G2, it is the third stage of cell cycle. In this stage, the cell has grown and the DNA has been replicated. This stage is preparing the cell for mitosis and meiosis. The last stage of the cell cycle is the M phase, which refers to mitosis. This is where the cell actually partitions the 2 copies of genetic material into 2 daughter cells. Cells use special proteins and checkpoints signaling systems to ensure that the cell cycle progresses properly. Checkpoints at the end of the G1 and at the beginning of G2 are designed to assess DNA for damage before and after S phase. If the DNA damage or abnormalities in spindle formation are detected at the checkpoints, the cell is forced to undergo programmed cell death or apoptosis. In every second of every hour and in every hour of every day, one of the most important events in our lives going on in our body is that cells are dividing. When cell divides, they make new cells. A single cell divides to make 2 cells, and these 2 cells then divide to make up 4 cells, and so on. This process is called cell division and cell reproduction, because new cells are formed when old cells divide. The ability of cells to divide is unique for all living organisms. Organisms grow because cells are dividing to produce more and more cells. In human bodies, nearly 2 trillion of cells divide in a day. Cell division depends on what type of cells. There are 2 ways of cell division, namely the mitosis and the meiosis. Mitosis cell division is how somatic or non-reproductive cell divide. Somatic cells make up most of our body's and organs, including our skin, muscles, lungs, and hair cells. In mitosis, the important thing to remember is that the daughter cells have the same chromosomes and DNA as the parent cell. Phases in mitosis cell division. The first phase of mitosis division is interphase. In interphase, the DNA in the cell is copied in preparation for cell division. This results in 2 identical full sets of chromosomes. During this phase, microtubules extend from these chromosomes. Second phase in mitosis is prophase. In this phase, the chromosomes condense into egg-shaped structures. Each chromosome is composed of 2 sister chromatids containing identical genetic information.
third phase is metaphase. During this phase, the chromosomes line up neatly end to end along center of the cell. The centrioles are now at the opposite poles of the cell with the mitotic spindle fibers extending from them. The last phase of mitosis cell division is telophase. During this phase, each pole of the cell, a full set of chromosomes gathered together. The single cell then pinches in the middle to form two separate daughter cells, each containing a full set of chromosomes within a nucleus. This process is known cytokinesis. Cells divide for many reasons. For example, our skin from our elbow. Cells divide to replace the old, dead, or damaged skin cells. Cells divide so living things can grow. Another type of cell division is the meiosis cell division. Meiosis is the other main way cells divide. Meiosis is a cell division that creates sex cells like female egg or male sperm cells. In meiosis, each new cell contains a unique set of genetic information. After meiosis, the sperm and egg cells can join to create a new organism. Meiosis is the reason why we have genetic diversity in all sexually reproducing organisms. During meiosis, a small portion of each chromosome breaks off and reattaches to another chromosome. This process is called crossing over or genetic recombination. The meiosis cell division has two stages, conveniently called the meiosis I and the meiosis II. In meiosis I, chromosomes in a diploid cell resegregate, producing 4 haploid daughter cells. It is the steps in meiosis that generates genetic diversity. Phases in meiosis I. During prophase I, DNA replication precedes the start of meiosis I. During this phase, homologous chromosomes pair and form synapses, a step unique to meiosis. The paired chromosomes are called bivalent and the formation of chiasmata caused by genetic recombination becomes apparent. In metaphase I, bivalents, each composed of 2 chromosomes, 4 chromatids, align at the metaphase plate. The orientation is random, with either parental homologue on the side. This means that there is a 50-50 chance for a daughter cell to get either the mother or father's homologue for each chromosome. During anaphase I, chiasmata separate. Chromosomes, each with 2 chromatids, move to separate poles. Each of the daughter cells is now haploid, with 23 chromosomes, but each chromosome has 2 chromatids. In telophase I, nuclear envelopes reform or cell may quickly start meiosis I. Cytokinesis is analogous to mitosis where 2 complete daughter cells form. This phase is preparation for meiosis II. Meiosis II is similar to mitosis. However, there is no S phase. The chromatids of each chromosome are no longer identical because of recombination. Meiosis II separates the chromatids produce cells, each with 23 chromosomes, haploid, and each chromosome has only 1 chromatid.
And that's it for cell cycle and cell division.